So welcome back friends, welcome back in another video tutorial from microbiology with Shagnik. So in this video tutorial we are going to see about the bright field microscopy. Although we have already discussed about a part of bright field microscopy in our previous video where we have discussed about the basics of microscopy. So the instrumentation part of the bright field microscopy we have already discussed, that is what are the various components in which the bright field microscope is made up of, right? What are the various part of the bright field microscopy? So today we are only going to discuss about the path of the light and what is the optical principle on which the bright field microscopy works and what are the advantages and disadvantages of the bright field microscope. So let's start. So here the light source that we are using, it is generally a sunlight or any LED light. Okay, so if you know about polychromatic light, then I hope that you know that the light coming from the sunlight or any kind of LED source is polychromatic. So what is the term polychromatic stands for? So polychromatic simply means it is a light source which consists of lights of various wavelength. Okay, so now the question is why we are using a polychromatic light, and what will be the equation, what will be the formula for the D in case of bright field microscopy. So the formula for D, that is here the D is the value that will help us to determine what will be the resolution of the microscope, what is the resolution power of the microscope. The formula for D in case of the bright field microscopy will be lambda average by 2Na. Okay, so now I want to discuss with you about why we are taking lambda average. So we have taken the lambda average because of we are using polychromatic light. That means in the light source there is a various lights of various wavelength. Okay, that is why we have to take the average when we are dealing with bright field microscopy. Okay, but if we are using any kind of monochrome as a light source, then the formula will be D is equal to lambda by 2Na. Okay, so now the question is why we are using a polychromatic light source, not monochromatic. So the answer of that question lies in its name. Remember it very carefully that we are discussing with bright field microscopy, right? So as its name suggests, bright field, that means we have to generate a too much amount of light to increase the brightness of the picture, right? Bright field microscopy commonly also known as light microscopy. That means here with the help of a too much amount of light we are generating a illuminated image, right? So that can be done with the help of polychromatic light. Why? Let's try to understand with a simple example. Suppose I told you to find out a small object in a dark room and I will only provide you a small torch. Okay, and in second case I will again told you to find out the object in the room, but I will give you 5 different light source. So what will be very preferable to you? Is it the case number 1 or the case number 2? So obviously the case number 2. So that is the case why we are using polychromatic light, because here light of various wavelengths are present. Okay, so that will help us to create a illuminated image. So now it is the most important advantage of our bright field microscopy, that bright field microscopy will provide you a illuminated bright image, but it is as well as a disadvantage too, especially when we are using bright field microscopy for analyzing biological specimen. Why? Let's discuss. So I want to discuss with you that the biological specimens are mainly semi-transparent in nature. Okay, semi-transparent. So what is the effect of biological sample being semi-transparent? So here the idea is, whenever you are putting the light, the biological sample will be illuminated with the field. As a result, it becomes very difficult to distinguish between the biological samples and the background. So that is why we cannot visualize, or it becomes very difficult to visualize specimens which is mainly biological specimens with the help of bright field microscopy. So why is this so? Let's discuss. Suppose this red marked circle is the field, okay, and inside this you have your biological samples, that is those red dots. Okay, now I am illuminating this. So let's see what will happen if you illuminate this in bright field microscopy. So you will end up with something like this. Are you are able to see what is in your field after the illumination? No. So that is what is going on in case of bright field microscopy. Although you will not going to see any kind of black background, instead of this you will see a white background, along with everything will white. That means the brightness is so large that you cannot separate what is your sample and what is your background. Okay, so now what to do? So we have to do what we have done here, meaning that we have to create a construct that will help us to distinguish between, or that will help us to find out our sample. Okay, so here what process we have applied to create the contrast? Yes, we have applied color. So that is what we have to do in case of bright field microscopy. That means we have to color our specimen. Okay, so we have various kind of chemical colors that can color the specimen of many samples. Okay, so those chemical colors are known as stains. So we have to use stains to color biological specimens before using the bright field microscopy. Okay, so as stain is a chemical agent, it will in turn cause a problem. What is the problem? So this staining process facilitates the death of the organism, because as it is chemical, it will surely harm and damage the cell. There is no doubt about this. So that is the major disadvantage of bright field microscopy. That means with the help of bright field microscopy, you cannot visualize any live biological samples. For visualizing biological samples, you have to stain them. Otherwise, you cannot create a construct that will help you to find out what is your sample. And if you want to create a contrast, you have to color them, right? And we have stains as their coloring agent, and which is chemical coloring agent, which cause disruption of the cells, and as a result we are visualizing only dead cells. So that is the major disadvantage and drawback of bright field microscopy. and this problem we are going to solve in our next video, that is the dark field microscopy. Okay, where we are changing the illumination that here we have used to create a contrast. So we will see in our next video how we will going to solve this problem, that is how we can see live bacterial or any kind of biological samples. Okay, so this was the path of the light in case of bright field microscopy. So here light is going on, it is concentrated by condenser, then it is imparted into the sample, from sample it is going into the objective lens. Suppose we are using a 40x magnification objective lens, then that means it is magnified 40 times with the help of objective lens, then it will fall on the eyepiece lens. Suppose we have a magnification of 10x in eyepiece lens, so 40 and 10 together will generate a magnification of 400x, that is observed by our eye. Okay, so that is the path of the light, and that is how bright field microscopy works. So advantage is it provides you a very bright illuminated image, there is no doubt about this, but disadvantage is you cannot visualize any kind of live sample with the help of bright field microscopy. Okay, because you have to stain them to create a contrast that will help you to distinguish which is your biological sample and which is your just background. So that was the concept of bright field microscopy. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends and subscribe to my channel to get more videos like that. So bye bye, take care.